Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goeie. I will be the Prorector and the Chair of the Degree Committee. First of all, I welcome Mrs. Kirthana Kunikullaya, and she will defend her academic thesis in public. And the thesis is entitled Short-Term Impact of Anthropogenic Environment on Neuroplasticity, a Study Among Human and Animal Models. Welcome all members of the Degree Committee, in particular the three supervisors. Two of them are sitting here and one is present online. And the supervisors are Professor Harry Steinbusch. He is Professor of Cellular and Translational Neurosciences at Maastricht University. Professor Thierry Cherlier, he is Professor of Neuroendocrinology at the Research Institute for Environmental and Occupational Health of the University of Rennes in France. And Dr. Jody Pawlowski, she is present online. And she is affiliated with the Institut de Recherche en Santé, and the institute is called Research Institute for Environmental and Occupational Health at the University of Rennes in France. And I will introduce the four opponents later during the ceremony. Welcome to the audience here in the aula, and welcome all followers of the live stream. Mrs. Kirthana Kunikullaya, may I invite you to give a summary of your thesis, and I wish you success in the coming hour. The floor is yours.
Thank you, dear Prorector. Dear Prorector, dear Corona members, and dear colleagues, friends, and family. I'm here to present a short summary of my thesis titled Short-Term Impact of Anthropogenic Environment on Neuroplasticity, a Study Among Humans and Animals. Since the main theme of my thesis was neuroplasticity, let me first introduce neuroplasticity. Plasticity is derived from the capacity of any material to mold itself into different structures and thus deliver different functions. In a similar way, brain has the capability to adapt itself structurally and functionally in order to deliver different functions. To give a simple example of neuroplasticity, I have taken the example of dart practice. When a person begins to practice dart, he often hits more off the target than on the target. But with time, with several repetitions and experience, the person starts hitting more on the target. This is because the areas between the brain responsible for this function is now structurally and functionally delivering in a better way, and this is what is called as neuroplasticity. The mechanism of neuroplasticity starts with a process called as neurogenesis, which is formation of new neuronal cells and glial cells from the neural stem cells. This process starts prenatally in the womb of the mother, and it continues through childhood, at least until adolescence. Thus, and this happens in most of the regions of the brain, and thus this period is often called the critical period because any insult during this time results in a negative impact on neuroplasticity. Other than neurogenesis, other mechanisms also play a role in neuroplasticity, such as strengthening of the synaptic connections, which is the connections between two neuronal cells, as well as pruning of those connections which are no more used. There are also several molecular changes, such as the hormones, receptors, chemical messengers inside the neurons, which modify themselves, and these are all the different mechanisms through which neuroplasticity occurs. One thing is that with time, with aging, neural stem cell pool goes on reducing in the brain, and thus, with aging, neuroplasticity reduces, though the process of synaptic connections or the molecular changes inside the neurons keeps continuing even during adulthood. However, the good news is that in adults, there is a pool of quiescent neuronal stem cells which can still divide in case of necessity, delivering neuroplasticity. Some of the areas which have been discovered for this process are hippocampus, hypothalamus, amygdala, cerebellum, cerebral cortex, and various such regions are constantly being discovered. Another interesting part about these areas is that they are integrated and also they are interconnected via the limbic system, and they are also connected to the peripheral nervous system, the autonomic nervous system, via the hypothalamus. And in this way, it can modify the heart rate or the blood pressure, which is one of the functions of autonomic nervous system. And this happens by the connections of these neuroplastic areas via the hypothalamus through the autonomic nervous system. Other than the autonomic nervous system functioning, we can also study the neuroplastic processes through tissue analysis of the brain in the animals or the human beings, which has been donated for science. We can also study the gene expression or the protein expression in the tissues and the cells. We can also study the behavioral change, such as artistic practice, or we can do brain imaging analysis in order to see what are the different areas that are stimulated because of the process of neuroplasticity. We can also record the electroencephalogram, which is the brain wave changes or the power wave changes in the brain through electroencephalogram. Coming to the factors affecting neuroplasticity, starting from as simple as diet, sleep, or physical activity, various other practices such as involvement in artistic practice, enrichment activities, or cognitively stimulating exercises, they're all said to positively impact neuroplasticity, whereas drug abuse, or chemical exposure is said to negatively impact neuroplasticity. The next concept that was studied in my thesis was anthropogene and exposome. Anthropogene is any environmental change caused or influenced by people, whereas exposome is all the factors to which a person is exposed to, starting from the prenatal period throughout the lifetime of the individual, almost until the death of the person, and these factors interact with the gene of the person to impact his or her health. To name a few factors which behave as exposome, it starts from the socioeconomic factors, air, water, sound we are exposed to, drugs and chemical we are exposed to, or the biological factors such as virus, bacteria, as well as lifestyle factors such as sleep, diet, again, and traumatic injury or stress, as well as enriching practices such as artistic practice. You will agree with me if I say now that several of these factors are the common factors which impact neuroplasticity. Thus, it was interesting for me to study one or two of these factors as part of my thesis, and I chose to study one potentially positively stimulating factor of neuroplasticity, music, on human beings. At the same time, in order to understand the molecular changes in the brain and with respect to the neuroplastic areas and the autonomic functions, I tried to study a chemical neonicotinoid using animals. First, music. Music and its effect on the brain has been studied several times now, and it has been shown that not just the auditory cortex, but several regions of the brain, including emotional centers such as the hippocampus, hypothalamus, cerebellum, the prefrontal cortex, as well as the somatosensory cortex, the motor cortex, several regions of the brain integrate together when a person is perceiving music or producing music. However, music is a difficult concept to study because it is made up of several features such as the tempo, percussion, or the genre of music, whether it's Western music, Indian music the person is listening to, or the pitch and the frequency at which the music is being played, the tones or the combination of the notes in a particular melody, or the timbre, which is the quality of the instrumental sound, or the lyrical component in music. Of these different features, I chose to study modes or combination of notes, and in that, I chose to study modes of Indian music. To give a brief example of what exactly the mode is, which is also called as raga in Indian music, I have taken a simple example of C major here, and this is how it sounds. So we see that there is a combination of notes, which is what is called as C major scale. It is also called as Raga Bilawal or Raga Shankarabharana in Indian music. Like this, I chose to study three different ragas or modes of Indian music, Raga Miyaki Todi, Malkaunts, and Puriya, which were played on Bansuri instrument or the flute instrument against a background of Tanpura, which is the drone instrument. We ensured that there was no lyrical component and no percussion instruments while intervening with this music. To give an example of the music which was played as an intervention in my research was This was just a very small clipping of raga mal counts, which was one of the modes we used in our intervention. Music such as these was played for 10 minutes during the intervention. However, before the intervention, during as well as after the intervention, we recorded several physiological parameters such as the anxiety levels of the individuals, stress levels via salivary stress markers, the blood pressure, heart rate variability, which is one of the index of autonomic function, which was done recording lead to electrocardiogram, as well as electroencephalogram was recorded using a 19-channel EEG machine. The findings of this work, which was presented in chapter 2 and 3 of my thesis, was that all the 3 music interventions led to a reduction in anxiety levels. Heart rate variability, it was different with respect to the mode, while Raga Miyaki Todi and Puriya led to an arousal response during the intervention when the person was listening to the music. After the intervention, there was sustained rise in parasympathetic parameters indicating relaxation. Whereas with Raga Mal counts as well as the control group, we found they had a sustained relaxation throughout the intervention, whether it's during or after. Coming to the electroencephalogram findings, we found that Raga Miyaki Todi led to brain power wave changes, which probably indicated that the person was having higher attention after the intervention. Raga Puriya indicated that they were probably having higher autobiographical recall or memory processes in the brain, whereas Raga Mal counts indicated that the person was trying to compare the features of music with the internal thoughts, probably due to the higher gamma activity during the intervention. Thus, we showed that music led to short-term changes in autonomic function as well as short-term changes in the brain power waves as recorded through electroencephalogram. The next concept studied in my thesis was the chemical insecticide thiacloprid, which belongs to a group of insecticides called as neonicotinoids. Neonicotinoids, as well as thiacloprid, they are usually sprayed on the plants or coated around the seeds and put in the soil, as well as they are injected into the trees in order to avoid the pests and the insects. However, initially, it was thought that it was not toxic to the non-target vertebrates and invertebrates. With time, it was realized that this chemical is absorbed systemically into the plants. While it is destroying the insects or the pests which are invading the plants, it is at the same time entering the food chain through the fruits and the vegetables, and it does not go off by simple washing. There is also soil that is run off into the aquatic bodies nearby, and thus affecting the aquatic animals, as well as the seeds are eaten by either rodents or the birds, thus affecting them as well. In my thesis, we asked the question, what is the effect of developmental exposure to thiacloprid on neuroplasticity? For this work, I chose to study zebrafish and rodents. In zebrafish, we exposed the embryos of zebrafish to different concentrations of thiacloprid until about 6 days post-fertilization, when the larval tissue was analyzed. Whereas in mice, pregnant mice were exposed between embryonic day 6 to 15 to thiacloprid at different doses, and later tissues from the amygdala, hippocampus, hypothalamus, and cerebellum was used for the analysis for their gene expression change in neurogenesis, neurotransmission, and hormonal pathways. The findings of this work, which was presented in chapter 4 of my thesis, was that the larval zebrafish, there was no change in neuroplasticity, whereas in mice, we observed dose-dependent, sex-dependent, and region-dependent changes in gene expression of neurogenesis, neurotransmission, as well as the hormonal pathways. Thus, we observed that exposure to thiacloprid does lead to a neuroplastic change depending upon the species that was exposed to. To summarize my work in the thesis, we saw that thiacloprid did lead to altered neuroplastic gene expression in mice, and it was dependent on the dose, the region, as well as the sex, whereas listening to music led to short-term autonomic and functional neuroplastic changes depending on the mode the person was listening to. Future studies may try to see what are the long-term changes of exposure to be it music or other enrichment modalities or chemicals such as thiacloprid and other neonicotinoids and their effect on neuroplasticity, not just at molecular level, but also the behavioral modifications. Future studies may also try to see if music and other enrichment modalities which have been shown to improve neuroplasticity, can it be used to improve the neuroplasticity once chemical exposure has been done? Also, this thesis indicates the dire need for finding alternate plant treatment products in order to prevent the toxicity that is happening to the non-target vertebrates and invertebrates, because prevention is always better than cure. Thank you all. With this, I finish the summary of my thesis and I give the word back to the prorector.
Thank you very much for this clear summary of your thesis.
Thank you.
The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Peter Peters. He is professor in nanobiology at Maastricht University. Professor Peters.
Thank you very much, Mr. Prorector, for giving me a chance to ask questions to the candidates. But before I do so, I will be very happy to congratulate you with the work that you have done. It was really impressive to see such a long introduction, but also nice data in the manuscripts that you presented. And I myself am not an expert in this field, as you know. As a nanobiologist, we study proteins and how proteins work as a machinery in cells and also in the brain. But I would like to come to this point at the end, though, to see whether you have thought about these things. But before I do so, I'd like to tell you that already my father showed me experiments when I was a young boy of 9, 10 years old on the farm when he was milking the cows. And he noticed very clearly again and again that when he was milking the cows with music of Mozart and Beethoven, the cows behaved much better than when he was listening to the news. So we usually had in the morning classical music, and in the evening the cows were exposed to the news of the day. And my father systematically saw a really big difference. So that is a very clear point that he already observed. I myself have interest in music, and actually when I'm going to retire, which is next year, I have an interest in getting a master's degree in musicology. So I really read those chapters in particularly with great interest, and that's why I like to come to that now by asking you some critical questions. And even though I liked your work, I am here now to ask you some critical questions, and you understand that, of course. So, and I have a great respect, let me say that also up front, for different cultures, for Chinese and Indian and all kinds of different cultures, African music. And I have been listening my entire life to music from all over the world. So it was therefore extra interesting to listen to you. And you proposed in the chapter 2, which I would like to focus my questions, to different kind of ragas. And I was reading that in very much detail, but I thought I need to study this also by listening to them and wonder how I would behave and how my stress level would behave when I was listening to them. And when I noticed this, I realized that my stress level went up rather than down. And it went up for all the three, no matter what, if I had to listen to it for longer than half an hour. And I exposed this to my family, which are all people from the farm in the Netherlands with a different culture and background. And none of them really liked this music. And for all of them, it caused stress. So the exposome that you talked about was actually negative rather than positive. Could you please start by saying why you choose this Indian music and why you consider this with your cultural background and whether it is different in different parts of the world and whether you have evidence for that?
Okay, thank you, dear highly esteemed opponent, for your kind comments and questions. So the question is, certain ragas lead to increased stress levels. Am I right? Okay, it is true that certain ragas or certain music does result in increased stress, and this has been the contention in research works using music, especially how to choose the music for intervening, whether it should be experimenter selected or the participant preferred music. And recently, the skewing is more towards the participant selected or preferred music than experimenter selected music. When I began, it was like almost equally experimenter selected as well as participant selected. Both were there, and now it has been shown in several research papers that the preferred music is much better. But if I have to say why I chose experimenter selected music, this was to create a uniformity in the intervention, because when we allow the participants to select a preferred music, we will not be able to study the music feature which we want to study and create a uniformity in the intervention, because not everyone is trained in classical music. If I ask them to choose a classical Indian music, for example, many in India would go with the Bollywood music or even Western music, if I ask them to choose, and there will be the lyrical component, and lyrics in itself has a different effect on the brain, or I'm sure even on the autonomic nervous system, and that is why I went for experimenter selected music.
Yeah, I got that point. You already indicated that nice in your introduction of today's presentation. But you didn't, let's say, discuss, for example, in the Western music, which I know much better, especially the classical music, that even if you listen to Bach or Beethoven that I mentioned earlier, or Mozart, that you have in there the rhythm that you already indicated. Did you have evidence that a largo, for example, or a slow beat is different than an allegretto?
Can you repeat? Slow beat and?
Fast beat.
Fast beat, okay. The tempo differences.
Yeah.
Yes. And the question is, sir,
whether you have noticed that there was differences in measurements, or didn't you study this?
I have not studied personally Western music until now. Coming to the tempo of the music, using even Bach, Beethoven, Mozart has been done before, be it the slow tempo and the fast tempo. And music has an effect on the autonomic functions, especially the heart rate and the respiratory rate, through a mechanism called as entrainment process. And they say the beat of about 50 beats per minute is considered a good tempo for maintaining the heart rate at around 60 beats per minute for the entrainment process. For the brain, when I read recent papers of fMRI, they say a beat level of about 120 beats per minute is good for the synchronization of the rhythm of the brain waves with the music. So there is no fixed beat rhythm which has been told that this is good for the health of the person so far, but then the entrainment process is there. Fast tempo leads to higher heart rate, this has been shown, and the slow tempo results in a lower heart rate and moderation of the heart rate. This is there.
Yeah. I dance twice a week tango, and in the tango you have Argentine tango, you have the slow version, the real romantic tango, and you have the milonga, which is the fast beat. And you can tell that 100 people in a room have a complete different atmosphere during the slow versus the high beat. And I'm sure that many proteins get expressed when you expose people to music if they like it. And that is where I want to come now, because you probably thought about the idea of finding out what, at the level of nanobiology in the cell, what proteins get expressed and which proteins are relevant for happiness and for joy, because that would be then the key to, let's say, better treatment, and especially also, which I didn't mention earlier, but people with Alzheimer and Parkinson, they enjoy music very much in their last years of their lives. And so it is very important to go into the nitty-gritty details and maybe even find drugs that express these proteins, so that also when you have no music around you, you still feel this happiness. So did you consider doing an RNA-seq and study the brain of, I mentioned my father's cows, whether you can choose another animal, I don't know, but the cows for sure experience this happiness. And if you expose them for a few hours to good music, then extract tissue and study the differences in RNA expression and see which proteins uniquely get expressed higher or lower depending on happiness or no happiness, or high beat, low beat. And then you have proteins that and then study. And then you can see what you can overexpress them.
Okay, thank you for the question. There are a few things I have to say at this point of time. Coming to the brain, I have not done RNA-seq experiment in this thesis or my work or protein expression analysis, but based on the research papers, I can answer this question to a certain extent. One, it was shown by Salimpur et al. Way back in 2012 or 14, when she showed that dopamine release in the nucleus accumbens region, the core region, is increased when music-induced pleasure is seen on the fMRI. And this happens more often when there is the peak emotion to music or anticipation in the music. This was shown. Other than this, several other neurochemicals have been shown to affect the pleasure response to music, such as oxytocin, prolactin, including certain opioids, and it has been shown that immunity also improves, like the inflammatory markers come down, pro-inflammatory markers come down. Other than this, yes, of course, cortisol, corticotropin releasing hormone, is also shown to reduce. These are, in general, the hormones which have been shown so far. Recently, human blood was drawn before and after about 20 minutes of performance. And also, the same group of researchers did another study where they collected the blood before and after 20 minutes of Mozart music listening. And they studied the RNA expression, which are the microRNAs which are expressed in the blood of the person. And they observed about 6 of them to be upregulated, like microRNA 132, 23A, 23B, 25, and 361. And they observed that these are related to the neuroplastic process of reduction in apoptosis, healing from neuronal injury, and even hormonal pathways. Some down-regulation was seen in 378A microRNA and 16.2. So this is one study which I felt was very interesting for me, because that is where I actually wanted to continue some time back with my music research, that I wanted to see what are the molecular changes in a person when music is heard or performed. I hope I've answered.
Yes, you answered very well, and I'd like to thank you for this very nice feedback to my questions. And I would like to also congratulate you at the end of my presentation to your parents who helped you, bringing you to where you are right now during your upbringing, and hopefully they gave you lots of good music during your upbringing. That's why you probably did so well with this work. And so I give the work back to the pro-rector, and thank you for your answers.
Thank you so much.
Thank you very much. Thank you very much, Professor Peters. The opposition will be continued by Dr. Xavier Coumoul. He is professor of biochemistry and toxicology at the University Paris-Cité in Paris, France. Professor Coumoul.
Hello, can you hear me well? I think so, because I have some feedback from my own voice, which is not.
Go ahead, please.
Yes, yeah, okay. So my voice is definitely music. So I'm even for that. I hope the opposition will be nice for you. I have a few questions, something like 15 questions, but some of them could, be answered, I guess, quickly. Some of them could be answered. You could take your time. And I have three different types of questions, one regarding the sources and the stimuli, one regarding the models or even the persons, one regarding the mechanism. So they are quite balanced for that, because I have something like 3 to 4 questions in each category. But before that, I'd like to thank you for your invitation. And also, I'd like to thank the jury for the nice interaction we have. Thanks a lot to Thierry, that I know very well. It's always a pleasure to interact with you. And your thesis was the subject was definitely very interesting, because I'm also a music lover, but I'm going from Baroque music to metal music. I'm listening to mostly every type of music, including international music. So starting with the sources, if you have the opportunity to do some similar work, what other type of music will you study?
Sure. Okay. Thank you, dear highly esteemed opponent, for your kind comments and questions. Coming to the question, what is the other type of music which I could have used if I'm going to intervene again? There are okay, the first idea which I have is, I have chosen 3 modes which are totally different with respect to the notes within them. If I may show the this way, we have a combination of minors and majors in the 3 modes. One of the ideas which I plan to have is choosing the second raga here, such as raga mal counts, which has a combination of perfect unison, minor third, perfect fourth, minor sixth, and minor seventh. The next mode which would have been ideal would be just addition of 1 more note to this. And the third mode would be addition of 2 more notes to this combination of notes of the pentatonic scale. So in that way, I would have known what is the difference exactly between by addition of 1 more note and 1 more note across the 3 modes. That's one thought I have. The second thought I have is with the instrument which I used and the control group which I used. I used a control group which had predominant silence throughout the intervention, and once in 2 minutes a little bit of natural sounds. One other control group I would have planned in my next study would be just playing the tanpura or the drone instrument. In the other 3 groups where we are intervening with bansuri or flute as an additional instrument, and one control group where there is only the drone instrument being played in the background. So this would have been a good control group to have along with the 4 groups I had.
Okay, thank you. Regarding the way we are listening music now, we are using, earphones, and they can silence the environment, the sound environment from outside. So do you think that the way we are listening music could change your results and eventually impact the type of biomarkers you have characterized?
Yes, I agree with the point that the way we are listening to music can change. It depends on the loudness level that we expose the person to music. Like a recent study showed that in recent generation who are constantly using earphones to listen to music of different types, they are noticing a higher degree of hearing loss is one thing which I have read about. So that's the thought I have about this.
Yes, I think it will be interesting to see what could happen indeed if you listen music, with space or we in a very restricted environment. Of course, regarding some of the models, it might be not very easy, but it's an important point. Regarding the pesticides, could you go back on how did you pick up those 2 pesticides? Basically, was it because of epidemiological evidence linking such pesticide to any kind of interference in neurodevelopmental processes, or any other type of ideas?
Can you repeat the question to your opponent?
Question was, what 2 pesticides?
Pesticides is the question. Am I right? For developmental exposure.
Yeah.
Okay, it was shown in 2017, one of the important papers from the European Union came out that neonicotinoids also result in developmental neurotoxicity. And one more paper in 2017 by Michel said that, in fact, they studied about 178 honey samples from across the world, and they observed the neonicotinoid being present in at least 80% of the honey samples from across the world. And it was more so in North America, Europe, and Asia, and less in South America. This was one good study to start with, wherein it was shown that in Europe, thiacloprid was maximally used compared to other neonicotinoids, and this paper was published in Science magazine. And that's how we chose thiacloprid, and not many studies had done thiacloprid and their effect on developmental neurotoxicity then. though there were studies which had already done imidacloprid as one of the chemicals to be studied for developmental neurotoxicity.
Thank you. Regarding the models or the person, so therefore the person who received the stimuli, we all know that the effect of music depends on the mood of a person. For example, if this person is tired or not, how would this affect your results? Did you think about, studying the music in different?
It does affect the mood of the person. When a person is in sad mood, they would prefer to use more of preferred music than experimenter selected music. That's one for sure. In fact, a recent paper by Irola also showed that people who are in a sad mood, when they listen to a preferred music of their choice, experience pleasurable experience while listening to the music, and it was shown through different brain regions which are helping in this pleasurable experience even when there is a sad mood. But in a lab, if the person's mood is not fine and I'm trying to intervene with music, I'm sure it won't have a good effect or the effect which would be ideal to be seen. Like we may find a difference in the anxiety levels, like we found a reduction in anxiety levels with our intervention, the 3 interventions music. But if the mood of people is not fine, I'm sure inter-individual variability will be higher if different people moods are different. For that, one thing we did in our study was we called them in the early morning hours when before their day started and after a good night's sleep, ensured they did not have coffee, tea before the intervention. So at least that way, they were all kind of happy, I think.
Okay. You also refer to person who are trained in music. And I guess that when you say that, of course, you can have different type of levels. Did you took it was in your manuscript. Did you took that in account for your study in terms of, I would say quartiles of training in music? And did you observe any kind of, effect regarding such kind of quartiles? I don't know if you did that, but could you imagine that there is an effect if you expand your research?
Yes, music training does have an effect on the physiological parameters when we record. We did record the musical training of the individuals who were in this study, all the participants, and we tried to analyze the data using ANOVA and trying to adjust for the musical training. And we also tried to see if music training as a separate subgroup analysis, if it has an effect, by categorizing those people with less than 1 year and more than 1 year of musical training. We did not find any significant differences, be it on our the autonomic function result, the chapter 2, or in chapter 3 as well. We tried to see if there is effect on the EEG. We did not find any significant changes. One reason which I can immediately think of why we did not see was they were trained in different types of music as well. It was not just Indian music they were trained. A few were trained with guitar, like Western music. Some were trained vocally. So it depends on that as well. But categorizing them into different subgroups would reduce the sample size, and statistically the power would reduce. So we did not further categorize them beyond less than 1 year and more than 1 year of training.
Okay. And did you try also, when you assess the effect of music, to manage the stimuli of other type of senses, eye detections of waves and things like that, the taste? Where did the person were in the same type of, environment regarding the other senses and not only hearing or listening?
All the recordings were done constantly in a single room where we would call one person per day only between 9 to 11 a.m. And all of them had the same set of electrodes, or be it the headphones. All of them had the same set. Just that with COVID, we would clean the apparatus every time, and sometimes it was difficult to recruit people during that time. Otherwise, all of them received the same type of intervention or the instruments.
Okay. Thank you. I guess I still have 2 or 3 minutes, something like that. Is that okay?
You have 1 minute left. Is that okay for a small question?
Okay. Yeah, small question. I will try to go fast, therefore. Regarding the saliva sampling, you did that also in you refer to that. Was the period also of saliva sampling, did you check before doing this saliva sampling, for example, if there was a better time to do that? Did you process to some kind of assess? Regarding cortisol also, you checked, you managed to measure cortisol. Did you think about measuring other type of biomarker, for example, of effects?
We measured cortisol and alpha amylase on the saliva sample we had, and this was based on the restricted funding we had, so I did not test any other biomarkers on the saliva samples. I did store them for a long time thinking I'll get some extra funding, but I did not test any other extra biomarkers. Was this the question?
Yeah, it was the question. It will be interesting to perform, I guess, omics on those saliva sampling to see if there's any other type of markers that could impact, could be impacted by regarding the difference between the mice, the zebrafish, and the effect of thiacloprid. I guess, is there any knowledge regarding the parameters which are named ADME, absorption, distribution, metabolism, expression of the molecule?
Thiacloprid metabolism in the mice or okay.
Explains the difference.
Give a short answer, please.
Yes, okay. In the zebrafish, I do not think there are metabolism studies which has been done in zebrafish. In mice, they have observed with imidacloprid and the metabolite of imidacloprid, and they have also tested that as a separate molecule and its effect on neurotoxicity. This is something which has been done. But thiacloprid on human beings, I have seen one study where they have used just one human being urine sample after giving a Noel dose of thiacloprid, and they have recorded 6 metabolites of thiacloprid in this person, which most of it was passed in the urine within 24 hours. About 2 metabolites was passed in the urine within 48 hours. It doesn't get stored in the, it doesn't get stored in the lipid tissue, so they did not find any storage in the adipose tissue, yes.
I propose to move to the next opponent.
Can I just ask one more question?
A small one, yes.
Yeah, you didn't refer too much to epigenetics in your manuscript, and do you think that music, if music was applied to a mice, for example, or a zebrafish, and you study, the next generation, but after, the second one, the third one, the fourth one, do you think that you could measure some kind of epigenetic changes?
Yes, I do think we can find some changes in mice, at least. I'm not very sure about what the zebrafish listens to, because I'm not very sure. Because it has both the vibration as well as the other sound stimuli, yes. Yes.
Okay. Thank you very much.
Thank you.
Thank you, Professor Kumul. The next opponent is Professor Bernadette Jansma. She is Professor of Cognitive Neuroscience of Language at Maastricht University. Professor Jansma.
Yeah, thank you, dear Pro-Rector. Dear candidate, first of all, let me congratulate you and also your supervisors to your great work. I read it with great interest, and I was very impressed by the broad scope of methods and skills that you show. It's really amazing what you do, not only humans and non-human animals, but also different types of methods, so it's really, really impressive. But given my expertise, I'd like to focus on chapter 3. It's the EEG study where you expose people to 3 types of music. So you also have a control group who did silent with bird songs in between. So you have 4 groups basically, and you did a pre, during, post intervention study. And you focus on power and also the intersubject correlation index, which is interesting to to study, because I know that a lot from social neuroscience, so it's great. And I just have questions, so and I also don't have a better answer, so like I shoot, you shoot back, right? So you already mentioned in the discussion about a certain potential limitation with regard to the stimuli, and it is also, I think, shortly addressed by colleague Peters. You very nicely describe the frequency specs of your 3 tunes. Like, I think, I'm not an Indian music expert, but it's pentatonic, hexatonic, and heptatonic, right? Colleague Peters referred to the cultural differences, like Indian ears obviously like that. For Western, it's disharmonic. The audio cortex might shoot, saying, "Ugh, I don't like that." That's actually why Peter got the stress reaction, I think. But coming from language, we also know that other characteristics in the stimuli might entrain the brain. And in your discussion, you shortly touched that point, so you're not totally neglecting it, but I would like to dig a bit deeper in there. So when I expose people to language, and I, for example, give a rhythm of 4 hertz per syllable, I will see an entrainment coming up of 4 hertz per syllable. Then I will see a 2 hertz for the word, and I will see a 1 hertz for the noun phrase and half a hertz for the sentences. So speaking to a hierarchical cognitive processing of the sentence structure. You study plasticity being entrained by music. The structure of your music is probably very similarly complex than sentence. Would you have the chance to look into more details of the rhythmic structure of your 3 tunes? And if you would, what would you expect? Could that explain the differences you find across alpha and delta and eventually gamma in the EEG, power as well as, yeah?
Okay, thank you, dear highly esteemed opponent, for your kind comments and questions. The question is, have I studied or will I try to see what is the rhythmic structure of my 3 Indian music interventions that we have used? The first thing is, what we played as the Indian music intervention did not have a fixed rhythm or a fixed tempo, because this is the beginning portion of Indian classical music called as alap, where the player elaborates the notes in a particular given scale, slowly, note by note, starting from the low notes, and slowly it is elaborated to reach the high notes, and then the scale is moved around, like they move around the scale in a very slow way. There is no fixed rhythmic structure in this music, but one thing which I consider as a limitation of my work, it is also to do that I do not know personally MATLAB application. So I would strongly want to study what are the music features within the music which I have used, such as the dynamic contrast. Though it is a very slow music, there is a certain degree of contrast and the phase change that happens, and the frequency of occurrence of notes is different in the 3 music interventions, such as in Indian music especially, every mode is said to have 1 or 2 notes which are said to be the king notes of that particular music, and that note is repeatedly played across the scale, be it in alap or be it in the whole song which is played after the alap. So frequency of occurrence of notes has been studied, I think, even in language, though I'm not at all an expert in language, but frequency of occurrence of nouns or words has been studied. Even in music, frequency of occurrence of notes was recently studied in 2015 using Indian music by a researcher in North Delhi, and they observed that the occurrence of major 2nd and major 6th was having a calm emotion in comparison to the minor notes of 2nd and the 6th, which was resulting in a sad emotion in about 120 participants which they studied. This was a very good study, and based on that, I actually wanted to study the music features, but somehow because I am not an expert in MIR analysis using MATLAB, it was difficult for me to do that into the study, but I think it will have.
If you find a colleague who could, you could do the spectrogram of the features and then correlate that with I think it would be very rich to see what goes on. Because related to that, not only do we have this input-driven entrainment in the brain, but we are also very good in predicting, so-called predictive coding. So if in language again, if I say, oh, the pizza is too hot to drink, what all happens to you here is a so-called ERP N400, also driven by theta and gamma, saying, oh, what is she telling? I didn't anticipate that. It's a violation of my anticipation, how this sentence should end. So I wonder whether in your music types, the prediction of what you just so nicely described in terms of core notes that repeat, do the listener have capabilities to predict? Is the prediction value across the three tunes similar, controlled for? Could you control or double check that afterwards and reanalyze based on predictive coding?
Yes, I think that's a very good question and a comment, and that is something which I wrote down, in fact, for Raga Malcounts, which was the second mode in our intervention, which is a pentatonic scale, and often it is people who are not so much trained in music, they relate a lot with pentatonic scale, and I still feel the higher degree of C2 which we observed with the second raga, which remained even after the intervention, was probably because of this attention which was there to predict what is the next note, because it just has 5 notes in it, and that too it already has a perfect, the first and the fourth, which is perfect, so there's no variation. So there is just 3 notes which are varying, which is easy for people to predict. I think, yes, that can have an effect, and it will be good to study in future for sure.
Yeah.
With my work, we haven't been able to study that.
Do I have time for 1 or 2 more? Okay. One more? Oh, yeah, then I have to pick, because I have 2 very...
if it takes too much time,
it's okay.
2 is fine.
So one question I have relates to figure 7. That's a great figure. It's on page 155. It's your basically result, the sculpt distribution and spectral pattern in your components.
Yes.
And then I see the component time course, and I'm really thankful that you plot that, because not everyone plots them, because you see it's like looking into the mind via a window which has temporal dynamics. What you see here is basically in the midpoint, like in the first panel in B, you have alpha, and in the C components you always have this, I would say, theta. You refer to it as delta, but it's around 5 Hertz. It's much closer to memory processing then. And you have a combination of peaks there in alpha and theta. You see that?
The C3 component.
Yes.
Yes.
And you already mentioned in your introduction that might be a pattern is related to memory processing. And if I think of Foster, who says, well, we have the perception action cycle, and that's how we try to make sense of the world we see. We always check what we see with the memory that operates via theta, and alpha is gating it. So what you see here is this beautiful interaction, and I would say a cross-frequency coupling between theta and alpha. Did you look into that? Do you have any thoughts on this cross-frequency coupling that is clearly visible in your data?
The C3 component. Yes, I commented in my slides that Ragapuria, which is one of the 3rd modes, is resulting probably in autobiographical memory process, and that is one mode which had a slight effect with the C3 intersubject correlation score. That was the only raga during intervention which had a intersubject correlation score, as well as that was the raga which the subjects recorded as maximally reducing the anxiety levels. it is probable, like I cannot clearly say whether it's the memory process or the theta or the alpha, because our data was collected on a 19-channel EEG.
No problem. I think you can spend another 10 years to figure this out. But I think my time is up. Thanks a lot. Yeah.
Thank you so much.
Thank you very much, Professor Jansma. The opposition will be continued by Dr. Marina Manet-Damas, and she is postdoc in neuroimmunology at Maastricht University. Dr. Damas.
Thank you, dear Prorector and dear candidate. First of all, congratulations for all your work, as well to your promotion team, and thanks for giving me the opportunity to be here as an opponent. I enjoy a lot reading your thesis. It was very interesting with all the music. I'm not that much into that topic. I'm more into the chapter 4. And that is where I have some questions, because I work in myasthenia gravis, which is a disorder that is, where the nicotinic acetylcholine receptor in most of the cases is affected by the presence of autoantibodies. So in your chapter 4, you investigated the effect of a pesticide that targets the nicotinic acetylcholine receptor, right? And you checked or studied the effect in development, neurodevelopmental, also in the offspring. So did you have any proof that this pesticide reached the brain and also passed, well, first passed the placental barrier and then the blood-brain barrier?
Yes, yes, there was pre-existing proof, not exactly with thiacloprid again. Imidacloprid, which is a more common neonicotinoid which is used, it was done in my study by Kimura Kuroda, group of workers, and they gave imidacloprid to the dams, and they observed what is the brain level in the pregnant dams itself of imidacloprid, as well as the F1 generation of mice, and again studied the developmental neurotoxicity in them. So in both of them, they did observe that there was a certain level of imidacloprid which is present in the brain, showing that it did cross the blood-brain barrier of be it the mother or the F1 generation mice. That's one thing. Other than this barrier, it is been shown that it can cross the intestinal barrier as well as the placental barrier, and it was observed even in the breast milk some time back. It was evidenced, so showing that it can cross different barriers.
Okay. Interesting. And then in your study, you measure mainly RNA expression, right, in brain tissue. But what about the protein levels in the brain or in the muscles where and other tissues where the AChR is expressed? Did you look for it, or do you think it will be reduced?
Yes, studying mRNA expression will give us only what may happen in future. So in this thesis work, we did not look at the protein expression levels, but it would be interesting, and definitely I'm sure certain proteins which affect neuroplasticity will be affected in these regions of the brain, because it has been shown that behavioral modifications are seen in the F1 generation mice after the pregnant mice are exposed to imidacloprid or clothianidin, and thus a change in protein is what would have led to a change in the behavior, is my thought about this.
And then there was one interesting finding that when you test different doses, right, you observe the opposite effect. So the 2 upper doses and versus the lower dose.
Yes.
So could you explain, like, why could that be? Is there any compensatory mechanism or protection effect?
Yes, not just thiacloprid. One point, a general point is several chemicals used in toxicology often show such dose response curves where it does not rise every time the dose is increased, or does not reduce together with the dose reduction. That's one point. Coming to the change which we observed in figure 3 and figure 4 of my thesis of that chapter, we observed that a dose of 0.6 milligrams per kg per day increased certain number of gene expressions in the hippocampal area, whereas the same gene expressions reduced with a much higher dosage. One reason for this, though it is not proven through my thesis, one reason could be desensitization of the receptors at this particular dosage, that most of the receptors are now occupied by thiacloprid, and after this, beyond this, even if we increase the dosage, there are no more receptors for it to occupy. Another probability, which I think is also to do with the cytotoxicity, that these cells are, maybe they have undergone apoptosis now, and thus there is no increase in gene expression with a much higher dosage. That is one thought I have. Coming to the other areas, such as the hypothalamus, in a similar way, we observed that at a higher dosage, most of the expressions reduced, except for in the amygdala, where there was opposite reaction as compared to the hippocampus. That's it.
Yeah. Okay. And then, because they have, in your study, they affect the chemical affects the acetylcholine receptor. So in your study, when the mice were alive, did you observe any locomotion problem? Because, for example, in the disorder that I study, patients suffer from muscle weakness and fatigue. So were these mice showing any kind of signs?
In our lab, we generally do not, we had not studied behavior with this experiment. The only thing we did was the tissue analysis, but other labs have shown a behavioral modification once the pregnant dams are given these neonicotinoids. Yes.
Okay, yeah, because I read a case report of a man that was exposed to this specific chemical and actually had transient myasthenia. I mean, it went away, but yeah, it was a case report. So I believe this could happen also to the animals. And also for the alpha subunit of the acetylcholine receptor, it has been associated with...
Yes, Mrs. Kirthana Kunikullaya, as you see, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense. And I would ask you to await our return with the results.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that.
Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Still ತೆಗ್ದ್ಯಾ? Theory. Theory ಹೋಗ್ತಾ ಇರೋದು ಅದು ಹುಡುಗಿನ ಗೊತ್ತೇ ಆಗಲ್ಲ ಆಗ್ತೀವಿ ಇದು ಬಂದಾದ್ಮೇಲೆ ನಮ್ಗೆ ಗೊತ್ತಾಗಿಲ್ಲ ಉಲ್ಟಾ ಮುಗಿ ಉಲ್ಟಾ ಮುಗಿ ಅಂತ ಆ್ಯಕ್ಚುಲಿ ಅದು ಗೊತ್ತೇ ಆಗಲ್ಲ ಆ ಥರ ಇದೆ ಅಂತ ನಮಗೆ ಯೂಟ್ಯೂಬ್ ಎಲ್ಲ ಉಲ್ಟಾ ಜನ
ಸಡನ್ನಾಗಿ ಬಂದು ನಾವು ಓಡೋದು
ಸರಿ ಇದೆಯಾ ತೆಗ್ದಿದ್ದು? Now you can come forward and take. ನೀವು ಹೇಳ್ಬೇಕು ಅವ್ರು ಬಂದಾಗ ಪುನಃ. ಅಪ್ಪಿ ಇಲ್ಲಿಂದ ಒಂದು ತೆಗಿಯಪ್ಪಿ ನಂದು, ಆಮೇಲೆ ಐ ಹೋಪ್ ದೇ ಡೋಂಟ್ ಲಾಕ್. ಸ್ಟೇ ಕ್ವೈಟ್ ನಾ ಬನ್ನಿ. ಸಾಕು ಹೋಗು.
Mrs. Kirthana Kunikullaya. Oh. I would, let me see where I have my text here. I would like to tell you that the degree committee here present has assessed the quality of your thesis and the quality of your defense. And in view of its positive verdict, and of course taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Steinbusch is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law, and invite now your supervisor to take the floor. Professor Steinbusch.
Okay. Dear Dr. Kunikullaya, do you promise to work in accordance with the principles of scientific integrity at all times, which means to be careful, to be honest, to be transparent, to be independent, and to be responsible? What is your answer to this?
Yes, I promise.
Thank you very much. Very well. So by the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Dr. I would say, Kirthana Kunikullaya, the degree of doctor, and grant you all rights attached to by custom and law of our university and the Netherlands. As evidence of this, I now present you with a degree certificate signed by the rector, who is here, signed by the secretary, and also signed by other members of the committee, and affixed with the official seal of our university. And I give this now to you. And the photographer can proceed. Just a moment. I open it correctly. This is the certificate. And I wish you all the best. All right. Let's have a look here. This is for you. It's not for me. It's for you. Okay? Please. Yes? Thank you. I would like to give now the word to Dr. Pawlowski, who will do a first short laudatio.
You can hear me okay? That's great. I'm just going to go ahead with a brief, I'd say a few words. So first of all, Kirthana, I want to say congratulations on your thesis and your thesis defense today. It's really well done, and I thoroughly enjoyed the discussion. And if I think back to when you started in the lab, I'm not sure doing a PhD was something you ever even thought about doing when you came to Rennes. And it was thanks to Harry during his visit to Rennes in October of 2021, I believe, that this idea of having your MD degree complemented with a PhD became actually a possibility. I know it took a few months to get all this sorted out, how this would go, but it's really great to see that you now have a PhD, and it's well deserved. I also wanted to say that your research is really interesting in that you used humans and rodents and zebrafish to join the breadth of your interest. And I personally have enjoyed seeing the research on how important and impactful music is on our well-being. And I'd have to say that because of this, I have been more conscious of the need for music in my life. I grew up doing lots of music, but I think sometimes reading your research has really made me conscious of how I need to put some music on to maybe decrease my stress levels. Music that I prefer, as you talked about today, and not even the music of others. It's something that I really find relaxing anyway, and how important that is in our life. I know that you have many more questions to answer in future research, and I look forward to seeing you answer these questions. It really has been a pleasure to be part of your thesis committee, and I wish you all the best in your future endeavors. Congratulations again.
And we move further to Dr. Chalier.
Thank you, Jody.
Thank you, Harry. Thank you, Prorector. Again, thank you very much, member of the committee. And congratulations, Kirthana. So Kirthana just joined the lab like less than 2 years ago, and it's very common. So Kirthana is a medical doctor. So she did not know much about the wet lab and basic research, but you turned out to be okay. See, we told you. So Kirthana contacted me during the pandemics, and she was working very often in the hospital in India with patients in a very stressful situation. And I know it's still very stressful. Now the stress will be reduced a little bit. She's done amazing work on Indian music that I did not know anything about. And then we thought about like making this project with animals, and we had some brainstorming about how to link your previous research with what I do in the lab. And again, I think it went okay. She learned quickly that the zebrafish is not very simple, that the techniques that usually work don't always work. But what amazed me is your motivation level. You never lose your motivation, and I really do appreciate this. You learn quickly very different techniques, so animal care, qPCR, immunofluorescence, the data analysis. We did not agree very often on some of the analysis, but we managed. And if I may say, one of the initial, the main issue that you had with the fluorescence microscopy was actually the chair was a little bit too high. So the feet were not touching the ground, and so she was complaining about this, like, could we do something about this instrument? And we thought about like getting you a little stool, but we never got anything. But again, what I like about your research and you is that you're really eager to share the information. You not only want to do the research, you also want to share the information with others, and so you have been teaching undergrad student in the lab, undergrad student in class, because I invited you in class and you really much enjoyed all of this. Altogether, I think you did a really great job, and I think you can do even more with what you have now. So you doctor twice, very much impressed by that. And I think you have tons of ideas. Just focus on a few that you can expand, not too many. There's a whole lot of research that can be done. just a few, and I think you'll do great. Congratulations, Kirthana.
Thank you so much.
Yes. So Kirthana, I also would like to congratulate you, and I also like to look a little back when we started this project, and basically it started in October 2021. It's not that long ago. But it started before that. I also was interested in Indian music, and I would like to refer to the words of Peter Peters. He mentioned that he was interested in Mozart, he was interested in tango, etc. I was interested in Indian music for a long time, and that has to do with the Beatles. And of course, when the Beatles went for the first time to India to listen there, and particularly George Harrison, and he looks to, I'm even emotional also, I don't know why. But he went there and he listened, and I was interested to see what is music doing with you. And I don't know, I did. I don't know why. Sorry.
Thank you very much, Harry. And again, thank you for your presentation. And I think we all enjoyed the discussion here. Again, thank you for the committee member here.
Esteemed Dr. Kirthana Kunikullaya, it's my great pleasure to congratulate you with your doctorate, and I do that also on behalf of Maastricht University and the Board of Deans of the university. And I would like to share some impressions of the committee with you. We heard and saw a very clear presentation of the subject of your studies. It was very interesting. And in your thesis, you have well done, precise studies, and you did research with adequate approaches and methods. And you also, it was based on a theoretical background, which I think is very important in these type of studies. And you have shown to be a very independent researcher. We liked your defense. You gave very clear, well-reflected answers, and we had an interesting discussion, a very surprising, interesting discussion, I think, for most of us. And you have shown that you are very knowledgeable in this new and interesting field of interaction of music and brain and its interaction and its impact. So, thank you very much, and I really congratulate you with that result. And I do also congratulate your 3 supervisors. And well, it has created also some emotions. And I would like to congratulate the 3 of you, Professor Steinbusch and Professor Charlier and Dr. Pawlowski. Congratulations with this result, that we have seen a very peculiar result and interesting studies.
Thank you so much.
And I hope it can be continued. I would like to include in my congratulations your family, of course, and friends and colleagues, and I think very few people are here in the aula, and I congratulate you warmly with this result. And I hope that the people of the live stream also feel obliged to what you have shown us today. So I wish you a very successful professional career and a happy personal life.
Thank you so much, dear Prorector.
I'd like to thank all the members of the degree committee, in particular the external members, for their input, for their critical reading of the thesis. Thank you very much for your input. Maastricht University appreciates your contributions. Before I close this academic ceremony, I would like to give the opportunity to the online opponents to congratulate the young doctor. And I would like to start, well, I would like to start again with you, Dr. Pawlowski. Maybe you want to say something personal in addition to your part of your laudatio.
I'm just going to say congratulations again, Kirthana. I'm sorry I'm not there to celebrate, but we can celebrate when you're back here in France. So enjoy the rest of your day. This is so well deserved, and I'm hoping you have a nice dinner and celebration plan tonight.
Thank you, Julie.
Thank you very much. Can I give the word to Professor Peters? Peter.
Thank you very much, Professor. I really like to congratulate you with this great presentation today and also with answering all those questions. I enjoyed the discussion very much, and I wish you a lot of success in your career. And if I'm retired next year and you want some critical person reading your work, you can always approach me if it is about music and science, because I have myself also emotions when I think about music, and I often have emotions when I'm in a concert hall, like Harry today, and that is the most of that is touching me in my heart, and your work contributes to that. Thank you.
Professor Kamul, the floor is yours.
Yes, I will not take 30 minutes or 45 minutes to congratulate you, because I have to be concise, and I'd like to congratulate you again, congratulate your supervisor. It's definitely a teamwork, but you definitely put your fingerprint on all this work. You have to know that your work is very original. There's not so many people who are studying at this point the positive aspect of the exposome, and music is part of it. Of course, it could be negative, as we discuss loudness, type of music, vulnerability of people. Of course, all this could be considered, but mostly, I guess everyone will agree that music would be always, most of the time positively considered as part of the exposome. So this is very important because it's original. And if it's original, you definitely have to use it also for your, further experience in life, jobs, but also on your personal side. Every time you will feel sad, according to your answer, you will know what you have to do. And finally, you also have to use this transdisciplinary work, which is also a key word, transdisciplinarity in your work, and the variety of models to also for your future personal and professional life. So I hope you the best for the future. And I'm really sad not to be here, but I hope to meet you one day, maybe in France, maybe somewhere else, maybe in India. I will love to come one day there. And all the best to you and to everyone in the room. Thank you.
Thank you so much.
Thank you very much. And I would like to give the last word to your one and only paranymph, Dr. Kai Zhang.
Hello, Kirthana. Congratulations for you. Yeah, very good speakers, and it's very better than me. Yes, and thank you for your take care in the real time. Yes, you take care of my life studying and many things. I also teach me my English and something. Very thank you. Yeah.
Okay.
Yeah, always my best friend.
Thank you.
Thank you very much. Please stay online. We are going to take a picture here. Here I close this academic ceremony.
Thank you.
Please stand